गुड मॉर्निंग स्टूडेंट्स। आज हम पढ़ेंगे फाइटोक्रोम के बारे में। फाइटोक्रोम एंड लाइट कंट्रोल ऑफ प्लांट डेवलपमेंट। Plants alfalfa and mung bean are germinated and grow in the dark, where they undergo a special kind of development called scotomorphogenesis. Scoto means dark and morphogenesis means development. Such etiolated seedlings have elongated stems and folded cotyledons, and they fail to accumulate chlorophyll. Now imagine these seedlings growing in soil rather than decorating your sandwich. Envision the elongating shoot pushing the delicate first leaves up through the soil using the apical hook to clear a path. When the seedling emerges from the soil and the limited energy reserves from the cotyledons or from the endosperm are exhausted, the seedling must begin making food for itself. This transition from scotomorphogenesis to photomorphogenesis is an extremely complex yet rapid process. Within minutes of applying a single flash of relatively dim light to a dark grown bean seedling, several developmental changes occur. First is a decrease in the rate of stem elongation. Second, the beginning of apical hook straightening. And third is initiation of the synthesis of pigments that are characteristics of green plants. The photochemical and biochemical properties of phytochrome. Phytochrome, a blue protein pigment with a molecular mass of about 125 kilodaltons, was not identified as a unique chemical species until 1959, mainly because of technical difficulties in isolating and purifying the proteins. However, many of the biological properties of phytochrome has been established earlier in studies of whole plants. The first clues regarding the role of phytochrome in plant development came from studies that began in the 1930s on red light induced morphogenic responses, especially seed germination. The list of such responses now includes one or more responses at almost every stage in the life cycle of a wide range of green plants. A key breakthrough in the history of phytochrome was the discovery that effects of red light on morphogenesis could be resolved by a subsequent irradiation with light of longer wavelengths called far red light. This phenomenon was first demonstrated in germinating seeds but was also observed in stem and leaf growth as well as in floral induction. The initial observation made in 1935 was that the germination of lettuce seeds is stimulated by red light and inhibited by far red light. But the real breakthrough came many years later in 1952 when lettuce seeds were exposed to alternating treatments of red and far red light. Nearly 100% of the seeds that received red light as the final treatment germinated. In seeds that received far red light as the final treatment, however, germination was strongly inhibited. Phytochrome can interconvert between phytochrome red and phytochrome far red. In dark or etiolated plants, phytochrome is present in a red light absorbing form, referred to as PR. This blue colored inactive form is converted by red light to a far red light absorbing form called PFR, which is a blue green PFR. In turn, can be converted back to PR by far red light. Known photoreversibility. This conversion or reconversion property is the most distinctive property of phytochrome, and it may be expressed in abbreviated form as follows. Red light convert PR into phytochrome far red, and phytochrome far red converted with far red light into PR. The interconversion of PR and PFR forms can be measured in vivo or in vitro. In fact, most of the spectral properties such as absorption spectrum and photoreversibility of carefully purified phytochrome measured in vitro are the same as those observed in vivo. It is important to note that phytochrome pool is never fully converted to the PFR and PR forms following red or far red irradiation because the absorption spectra of the PFR and PR forms overlap. Thus, when PR molecules are exposed to red light, most of them absorb the photons and are converted to PFR. But some of the PFR may also absorb the red light and is converted back to PR. The proportion of phytochrome in the PFR form after saturating irradiation by red light is only about 88%. Similarly, the very small amount of far red light absorbed by PR makes it impossible to convert PFR entirely to PR by broad spectrum far red light. Instead, an equilibrium of 98% PR and 2% PFR is achieved. This equilibrium is termed the photostationary state. In addition to absorbing red light, both forms of phytochrome absorb light in blue region of the spectrum. Therefore, phytochrome effects can be elicited also by blue light, which can convert PR to PFR and vice versa. Blue light responses can also result from the action of one or more specific blue light photoreceptors. Whether phytochrome is involved in a response to blue light is often determined by a test of the ability of far red light to reverse the response, since only phytochrome induced responses are reversed by far red light. PFR is the physiologically active form of phytochrome. Because phytochrome responses are induced by red light, hypothetically they could result from either the appearance of PFR or the disappearance of PR. In most cases, studied quantitative relationship holds between the magnitude of the physiological responses and the amount of PFR generated by light, but no much relationship holds between physiological responses and the loss of PR. Evidence such as this has led to the conclusion that PFR is a physiologically active form of phytochrome. The use of narrow wave band red and far red light was the central to the discovery and eventual isolation of phytochrome. However, a plant growing outdoors is never exposed to strictly red or far red light, as are plants used in lab-based photobiological experiments. In natural setting, plants are exposed to a much broader spectrum of light And it is under these conditions that phytochrome must work to regulate developmental processes, and it affects on both the quality and quantity of incident light reaching individual plants and leaves. Characteristics of phytochrome induced plants. The variety of different phytochrome responses in intact plant is extensive in terms of both the kinds of responses and the quantity of light needed to induce the responses. A survey of this variety will show how the diversely the effect of a single photo event, the absorption of light by PR, are manifested throughout the plant. For ease of discussion, the phytochrome induced responses may be logically grouped into two types: rapid biological biochemical events, and second is slower morphological changes, including movements and growths. Some of the early biochemical reactions affect later developmental responses. The nature of these early biochemical events, which compromise signal transduction pathways, will be treated in detail earlier in the chapter. So, thank you, students. It's completed.